Good morning, everyone. I'm very excited to be here this morning. I'm sorry that we can't be in person, but such is life, right? A big shout out to my team on Common Experiment 2. We've also been meeting monthly and have got our protocol together. We've had some very lively discussions about how to do this. This experiment that we're going to be doing is looking at the effect of cover crop termination timing on slugs, insect pests, diseases, weeds, and then cash crop performance. And so we've had a lot of variables to figure out, especially as we start collecting data. So I think that'll become evident as we move through. Next slide, please. So the states that are participating in Common Experiment 2 are those states there with the that are highlighted. And I'm in Iowa, so I'm going to be leading the plant pathology part of this experiment. And then we have John Tucker in Pennsylvania, right, John, I think. And he's going to be he's leading the insect part of this experiment, and then Colleen Chase down in Florida will be leading the weed science part of this experiment. And John and Colleen will both be presenting a slide as part of this presentation. So just to let the people in charge know that at some stage we're going to need to get John and Colleen on the video as well. Next slide, please. So what is the whole emphasis of this experiment? So as I mentioned, we're going to be delaying cover crop termination. And so cover crops, I showed this slide at our first meeting, but I just wanted to go over it a bit. So we know that cover crops have a lot of benefits, and farmers are interested in pushing cover crops to achieve those maximum benefits. And so getting that cover crop to grow as long as possible before they terminate the cash crop and before they terminate the cover crop and plant the cash crop. And then in some areas, like particularly up here in the Midwest, we can get very wet springs that come in, and so a farmer can't get into his field and delay terminating that cover crop, and so what happens then? So if you go back and look at the previous research, cover crops have been shown to be very effective at managing weeds, and so when you consider that herbicide-resistant weeds are becoming more and more of a problem, this is an important benefit from cover crops. We also know that cover crops can provide an alternative food source for slugs, and that means there's less damage to the cash crop. And then also cover crop biomass provides a habitat to beneficial invertebrates that may suppress slugs or other insect pests. So those are the positives of delaying cover crop termination, but there's always a but, right? But a rye cover crop has been shown to act as a green bridge for seedling disease pathogens. And so that means if we terminate the cover crop too close to planting the cash crop, then that can increase the chance of seedling disease. And what happens with seedling disease is it either kills the cash crop or it reduces the vigor of that cash crop, and that results in yield drag. Cover crops can also act as green bridges for insect pests that can damage the cash crop. And last of all, high cover crop biomass at planting may affect planting that cash crop, establishment, growth, and development of that cash crop. And so the purpose behind this experiment is that we're going to be trying to figure out how cover crop termination affects that cash crop, because at the end of the day, that farmer wants yield. Next slide, please. So our treatments are pretty simple. We have a no cover crop control, so that will just be bare soil. And then we have what we call a planting brown treatment, which is where the cover crop will be terminated using glyphosate 2 to 4 weeks before the cash crop is planted. And then we have the planting green treatment, which is where that cover crop is terminated after the cash crop is planted, so 7 to 10 days after that cash crop is planted. And these first 3 treatments are mandatory. Everybody will be doing those treatments. And then we have a 4th treatment, which is optional. And I want to say that 8 or 9 of the 15 sites that are taking part in this study will also be doing a planting green-brown treatment, and that is where the cover crop is terminated using glyphosate 3 to 7 days before that cash crop is planted. Next slide, please. This is our field protocol. So everyone is going to be using a cereal rye cover crop, and we're going to drill that and seed it at about 60 to 70 pounds an acre. Everybody will be planting the cash crop corn. When we first proposed this experiment, we were debating whether to use corn or soybean. But the reality is that there's not a whole lot of interesting things when it comes to diseases on soybean. And so we've moved more to corn. The other things that we have is that there'll be 50 pounds of nitrogen at planting, and then the balance of the nitrogen, depending on the site, will be side dressed at growth stage B4 to B6. Field history. All of these fields are going to be a previous crop of soybean and no-till. So we've had to, and obviously for between years, so just like Common Experiment 1, we'll be doing this in 3 years, starting in the fall this year, 2020. So we're following exactly that timeline that Hannah just showed you. And then we'll be alternating fields. So that we can get that previous crop of soybean. Up here in the more northern regions, we'll try to plant an early maturing soybean so that we can get that cover crop in a timely fashion so that we can get good establishment. Pesticides are obviously important because we're going to be looking at pests, right? We want to see insects, we want to see disease, and we want to see weeds. No insecticides are going to be applied to the field or to the seed. We will use fungicide seed treatments on our seed. We know from work here in Iowa that that doesn't impact too much how much seedling disease comes about. So there will be fungicide seed treatments. And then I know Carlene's probably going to talk a little bit more about this, but we will have a standard burn down and pre-herbicides, and then we'll have post-herbicides as need be. Next slide. Oh, not this slide, but the next slide. We have to be careful with where those herbicides are. Okay, thank you. Can we go back to the previous slide? So this is the previous, just kind of gives you an idea of our experiment. So it's a randomized complete block design with a split plot or strip plot arrangement that you'll see. And we have 5 to 6 reps. The brown there that you see on the green are the treatments, and then the brown, we need to have enough room between our plots to move machinery, especially for herbicides, etc. Next slide. So one of the things that we had to think about, because we needed to collect all this different data, is where are we going to collect this data? And so our plots are going to be at least 16 rows long. So that's what's shown here. So we'll have probably 4 planting passes. And then you can see that we have a subplot for weeds. So that's where we'll be collecting the weed data. And that's apart from where the disease subplots will be. So we'll collect disease data in there. And then we have the insects subplot where we'll be collecting the insect data and different rows where we'll collect data on slugs, and then also the predation data. And then we will collect the cash crop data shown there, and also we'll be collecting the cover crop data across those plots. Next slide, please. Okay, so just to highlight what different data we're going to be collecting. So this is the disease section, and I showed you on the previous slide that will be in a couple of rows in that big plot. So what we do is we go out and we assess seedling root rot at V3. So this is basically going out into the field, digging up some seedlings, carefully washing those seedlings, and then all participants will be given directions about how to take a photo of those seedlings, and then those photos will be uploaded into the awesome website so that Rochelle can then get at the photos. Rochelle is the postdoc in my lab working on this project, and she will then go in and assess the amount of root rot that she sees in all of these seedlings. And then another piece of data that we'll get is stalk rot, which will be at physiological maturity. It's pretty simple, basically just splitting the stalks of those plants and looking at how much of the plant is rotted. So our hypothesis is that with the planting brown, we will see limited seedling disease. It'll be similar to the no cover crop. But in the planting green brown and the planting green, that we will see seedling disease. We don't know whether we're going to see more in the planting green versus the green brown, but that will be up for debate. So next slide is insects. So we need to give the mic to John Tucker, please. And then we'll come back to me after Colleen.
Right, looks successful. Hi, Alison, thanks for leading into the insect stuff. I should say, acknowledge that the kind of the insect and slug piece kind of has complicated things. So that's why we have bigger than average plots, because insects move around, particularly the predators we're interested in move around. So the bigger plots just allows a little more room for them to move around and decreases the chances they're going to move among treatments. For the insect piece, it's pretty much 3 pieces. So we're going to evaluate caterpillar and slug damage at V3 and V5. And we're going to provide protocols on how to discriminate between caterpillar damage and slug damage. It's pretty straightforward. Of course, in parts of the world where slugs are not your concern, this won't be, this will basically be a caterpillar thing. And if there are other pests that are critical for your part of the world, we'll work with you to figure out what needs to be characterized. But the main pests that we see here in the mid-Atlantic region are black cutworm, armyworm, and slugs in no-till cover crop systems. And to more specifically look at slug populations, we're also going to use shingle traps, and these are just artificial shelters that slugs gather under, and they will be easily counted just by checking these things early in the morning. And then because the predators are a critical piece of this, for a while now, my lab and others around the country have been using sentinel prey assays. So these are more or less caterpillars that we pin to the ground and we leave for a certain amount of time and we come back and see if anything's eaten them. And this is just an assessment of the activity of the predator community. So whether it's ants or ground beetles, centipedes and the like, they'll attack those caterpillars, and we can use that as kind of an index of how active the predator communities are. And we'll provide detailed protocols on how to do this, how to make the shingle traps, which is pretty straightforward, how long the caterpillars need to be in the field, what the supplies are. But the data that we've been collecting in my lab from these 3 efforts provide pretty great insight on how cover crops are influencing pest populations. So I think that's all I was supposed to cover, Allison. So I guess I'm done. Off to Carlene.
Okay, so thank you, John. And so my area is focused basically on answering the question, what are the weed management implications of planting green? If we delay the termination of the cover crop, we expect to see that there's going to be far more biomass, and we want to understand how does this additional biomass and delaying that termination of the cover crop till after planting the corn, how does that influence the dominant weeds that we see? And also, does that influence weed density and also weed biomass? So those are the kinds of data that we want to collect in the cover crop just before termination. We want to determine how much biomass we have at that termination stage. We want to be able to understand what are the primary weeds that are occurring in the rye at that time and how many weeds that we have. And we'll be measuring, taking those measurements within quadrats. Then, because we are working with both the disease and the insect folks, who the collection of their data really is best done under conditions that are weed free. The majority of the plot that you saw will be managed so that it's weed free, apart from what we're calling the weed subplot. And in order to do that, what we're going to be doing, as you can see in the table down below, is that we are going to be using a cover crop burn down, which is going to, for the control, and the planting brown is going to occur as an early pre-plant treatment. And then it's going to happen just about 3 to 7 days for planting green brown. And for planting green, it happens actually after the cover crop is planted. In 3 of those cases, as you see, we're also going to utilize, where needed, pre-emergence burn down to make sure that the plots are clean of weeds going into the corn crop. So as Alison talked about, in our 4-row subplot, what we want to do is that where you see that pre-residual, where we're going to apply a residual herbicide to the field to allow us to continue to have the rest of the 12 rows weed free, we are going to keep 4 of those rows that are not treated at all with that pre-residual until the V4, V5 stage. And that will allow us to be able to collect the weed data that we need, that weed data that will allow us to understand whether having delayed termination, if we're seeing any kinds of weed shifts, or are we seeing any changes? Are we having a lower weed density and lower weed biomass? And because we have a group that's not necessarily weed scientists, we're trying to keep this really simple in terms of just getting total weed density and total weed biomass. For the folks, in terms of understanding the weed coverage is going to be the weed coverage of the dominant weed species. And so we are going to assist non-weed scientists to capture that data, not only by taking photos of each quadrat, but we're also going to have things like WhatsApp groups where they can share photos with us, the weed scientists, to assist with helping them determine what the dominant weed species are in their plots. After we collect those data in our subplots at that V4, V5 stage, then we would apply a herbicide to the weed plot so that for the rest of the season it remains weed free. And next slide, so that Alison could go back to the rest of the presentation.
Okay, thank you, Colleen, and thanks, John. That was awesome. So obviously we need to collect other data. So we'll be collecting data on the cover crop and also the corn. So that gives you an idea of the data that we'll be collecting, and Colleen talked about some of that. And then next slide. The next slide kind of shows our annual timeline of activities for Common Experiment 2. One thing that we had to agree on is when we were going to go out to the field. As it is, we're going to be going out to the field about approximately 11 to 12 times during the growing season to collect all this different biomass. It was up to about 18 times, but we came to some agreement about when we could collect different types of data together. And so that kind of tells you that, for example, you know, at V3, we're going to be going out and doing stand counts, growth stage. We're going to be collecting insect data. We're going to be collecting seedling disease data, and we're going to be collecting weed data all on that day when we go out there. So that's going to be a very busy day. Final slide is just a big thank you to the Common Experiment 2 team. Thank you all for your time, and I'd be happy, or we'd be happy to take any questions or comments when we're allowed to. And I trust that we are within our time spot.